Hi, thank you very much for selecting this abstract. So we're going to talk about the Rostro-caudal architecture of the frontal lobe in humans. So you may know about the functional model of the organization of the frontal lobe with the most advanced function localized in the most anterior part of the frontal lobe and simplest function being localized in the posterior part. It's been mentioned in the book of Marcel Mesulam, where you can see that you have idiotopic areas that are in the motor cortex, and then you have associative areas and very high associative areas in the most anterior part. It's been demonstrated by researchers using functional MRI, such as Etienne Coquelin, who use functional MRI to show that as you're going towards the frontal pole, you require more integrative power in terms of function. And then it's been reviewed in many papers. You may be familiar with review in Nature Review Neuroscience by Badre and Desposito on the hierarchical organization of the frontal lobe. And what we were interested about is trying to extract from magnetic resonance imaging some biomarker of this organization. So what we did, we used a previously published parcellation of the frontal lobe that has been based on the measure of structural connectivity in the frontal lobe. So those regions are separated based on the pattern of connection with the rest of the brain. It's a very highly reproducible result. And for example, here you can see on coronal section on the lower part that the left hemisphere is very similar to the right hemisphere. And you can also use a different data set and you obtain the same division. So we try to extract from those regions the functional pattern of activation by, so this is how we created those regions. We used the database of the Human Connectome project. 12 participants have been acquired with 270 directions on a multi-shell acquisition with a voxel size of 1.25 cubic millimeter. Our approach to quantify tractography was a two-fiber probabilistic cubal tractography, which is kind of like an old approach to do tractography, but that was the most reliable approach to obtain a parcellation. I can touch on that point later if you're interested. And then the way you construct your connectivity matrix to divide the brain is just to associate 2 by 2 regions of interest within the same hemisphere. Additionally, we use also anatomically defined, manually defined region of interest to compensate for some limitation, well-known limitation of tractography. So we took each of the regions and we ran it into Neurosynth, which is a famous database online where you can run large meta-analysis on all the previously published fMRI. And Neurosynth includes a function which is called the decoder. It's really fun. You can load your region of interest, and it's going to give you the probability of a specific function to be activated within your region of interest. We did that for the 12 regions, and we identified that we have a functional specificity of those regions. I don't know if the math is working. It's really good. So you can see here, the line that is here is all the 12 regions and their contribution, their likeliness to be activated for those functions. You can see that you have a clear specificity in the frontal lobe that can be characterized by the fact that those regions are defined by the structural connectivity. On the right matrix, we look at the coherence of the spatial distribution of the activation with the localization of the region. So that's a spatial correlation. Although it's not as good as the likeliness of being activated, you can still see that you have a very coherent specificity of those regions, which means that when you activate for phonological functions, you're more likely to activate within the region number 7 rather than neighboring regions. And those functions were organized as predicted with a posterior-anterior gradient of complexity starting from idiotopic area, which are foot and motor function. The next term was actually the hand area, to modality-specific association areas such as the frontal field, language production, pre-SMA, and phonological, to high-order association areas including semantic, social, and the appreciation of values. Our next step was to look at MR acquisition in this participant to try to collect information on anatomical information, for example, on cortical thickness. And cortical thickness derived from T1 indicated that you have an anterior-posterior gradient, with the most anterior region being the thicker and the posterior region being the thinner. And this was reproducible on the left and on the right hemisphere, and the lower portion you have the same result reproduced in 3 individual participants. However, cortical thickness is not necessarily the most reliable measure. Cortical thickness derived from T1 is not necessarily the most reliable measure of cortical thickness when you compare it to postmortem. And I learned this really the hard way. As you can see in the table, not only sometimes cortical thickness is overestimated, but it might be also underestimated, and you don't have a reliable distribution in this over and underestimation, which is a problem for intra-individual comparison between regions. So although we have a highly reproducible result on one hand, we cannot directly match it to a real true measure of cortical thickness. So we turn ourselves to other measures that also have limitations, such as T1, T2 quantification of myelin based on MRI. And this indicated that the most anterior regions actually seems to be the less myelinated compared to the posterior regions. A result that I interpret as the more you're going towards the frontal lobe, the more regions going to take their time to get their answer as an old wise man. Then we tried to look at whether there was some change related to the cytoarchitecture. And we have the chance today to have the big brain. You can spend hours looking at microscopic resolution of the cortex and look at the organization of the cells from the entire frontal lobe. And what we try to do is to quantify, so this brain has been stained with silver staining, we try to quantify how much the staining stains neuronal cell inside all those regions. And if you do so, you see that you have another gradient with posterior region composed of very big neuronal cells, not that much space together, giving this very dark contrast. And as you're going towards the frontal pole, you have smaller neuronal cells that seem to be really spaced. And this shows as being less stained by the silver. And again, this was reproducible in the left and in the right hemisphere. So we started thinking what might be between those neurons that can, be related to the complexity of the process in those regions. So I'm coming from the diffusion field. I looked at the mean diffusivity to see whether there were more diffusion of water inside the cortex in the regions. And again, so this is just to give you the idea of how diffusion-weighted imaging works. We're going to quantify how much molecule of water diffuse inside the tissue. And the mean diffusivity is the average amount of diffusion that happens. So the more the water, as freer the water, the more mean diffusivity you're going to have. And there we could see that as you're going towards the frontal pole, the more you're going to have water diffusing and moving inside your cortex. And this was interesting because we jump on it thinking, oh, maybe there are more dendrites and connections that explain the complexity of the processing. So we've been thinking a long time about that, how to try to assess the level of complexity and process that might happen inside the cortex. And we come out with an experimental solution, which is to measure the level of entropy inside the cortex derived from resting state functional MRI. So for those of you that are not familiar with entropy and cannot read complex equation like me, the way you can represent it to yourself is like you trying to measure the complexity of the signal. And if you're measuring the complexity of the music coming from a flute that has only 7, 8 tubes, you're going to have a melody that is much more simpler than if you're measuring from a flute that is much larger. The assumption is not that you have a flute inside the brain. The assumption is actually in the brain, when you have a bigger flute, you have more neurons and more connections that can process a given richer signal. Okay? And when you did that, we find strong correlation in the left and the right hemisphere, showing that the further you're going towards the frontal pole, the more complex the melody or the signal you're going to find. And this again was reproducible at the individual level and in the left and the right hemisphere. So the take-home message is, I think there is an anterior-posterior trend in the frontal lobe going from the C+ to the most advanced frontal functions. This has been demonstrated many times with many different ways in the past. This is not new, I just want to remind it. But what is interesting is this trend is characterized by an anatomical signature of structural connectivity, T1 derived cortical thickness, myelin density, water diffusion, and anticity of silver staining, as well as bold signal complexity at rest. And now that we have this model in the healthy brain, we can try to see whether there is any different way of having this gradient in pathological brain with a different maturational trajectory. Thank you very much.
The floor is now open for questions.
Hi. I was a little bit confused by the relationship that you saw with the cell density, where it seemed like the higher the cell density, the higher, so the cell density increases as we go towards the frontal pole?
No, it's the inverse because that's a staining. So the more staining you have, the less light you get coming out from the slice. So the higher it is, the lower is the staining.
Okay.
I should have mentioned that, that's right.
So then we have lower cell density in the frontal pole, and that would account for the increased mean diffusivity data.
It's like you have lower cell matter. You may have more cells, but they're smaller. So you see it less, it's stainless.
Okay, all right, great, thanks.
Yeah.
I'm just curious, is CBR1 the motor cortex?
So, I'm just going to come back to it. So it is, oh gosh, what did I do? Okay. There you go. So it's the motor cortex, but it extends a little bit more anteriorly.
And so on the, if you go back to the cell staining.
Yes. So I'm going to do it.
I'm wondering how you delineated the cortex, because it looks like you might have missed layers 5 and 6 on the motor cortex.
That might be possible, yeah. That's why I showed those snapshots, because you have the automatic extraction, which is based by the identification of gray versus white, that also has limitations. And then you have here just a display of the result from the cytoarchitectonic staining.
So given the limitations with the thickness measurements,
yes.
the MRI, and
yes.
possibly the density, how does that affect your interpretation of the complexity calculation?
You mean with the entropy?
Yes. So
yeah.
maybe the motor cortex is thick and low dense.
Well, that's a very good question. Well, you know, the fMRI resolution is not that good. It's like 2 millimeters. So it might have affected in terms that we don't have voxels that are in between white matter and gray matter in principle, because we underestimate the thickness of the cortex. So we should be in. But in the case that the complexity is different between the different cytoarchitectonic layers, and we underestimated the first layers, there might be a problem. I have no solution how to solve this one yet.
We have time for one last question.
I'm generally in sympathy with what you're doing, but I'm rather puzzled that you refer to the ratio of the intensity of a T1 weighted MR image to the intensity of something which is miscalled T2 weighted MR image, which is actually not T2 weighted.
Okay.
Why you call it a quantitative measure of myelin? It doesn't seem to be quantitative at all.
So this is a debate that doesn't concern me directly. We just do it because this was the only data that we have, but I appreciate your point. I think this question...
It's just a ratio.
Yeah, it's a ratio.
Two arbitrary images.
I will take care of correcting this.